Hello students, today in this video we are going to study Genomics, Human Genome Project and DNA Fingerprinting. Now let us study first point, Genomics. What is a genome? Genome is the total genetic constitution of an organism. म्हणजे आपल्या शरीरातील पेशीमध्ये 2n इज इक्वल टू 46 क्रोमोझोम आहेत. म्हणजेच 23 गुणसूत्रांच्या जोड्या आहेत. या 23 गुणसूत्रांमध्ये असलेल्या DNA किंवा जेनेटिक इन्फॉर्मेशनला आपण जीनोम असे म्हणतो. अल्टरनेटिव्हली इट इज अ complete copy of genetic information, that is DNA, or one complete set of chromosomes, monoploid or haploid, of an organism. The term genome was introduced by H. Winkler in 1920. Now, what is a genomics? Genomics is the study of genome. How this study is done? It is done through the analysis, sequencing and mapping of genes along with the study of their functions. The term genomics was coined by T.H. Roderick in 1986. There are two types of genomics: structural genomics and functional genomics. Structural genomics means it involves mapping, sequencing and analysis of genome, and functional genomics, it deals with the study of functions of all gene sequences and their expressions in organisms. Now let us study about the applications of genomics. आता genomicsचे कोणकोणते ॲप्लिकेशन्स आहेत ते आपण बघूया. फर्स्ट, इम्प्रूव्हमेंट ऑफ क्रॉप प्लांट, ह्यूमन हेल्थ अँड लाईव्हस्टॉक. नेक्स्ट, applied in a number of different sectors including medicine, biotechnology and social sciences. Next, treatment of genetic disorders through gene therapy. Then, development of transgenic crops having more desirable characters. For example, BT cotton, BT brinjal, etc. And it is also applied in forensic analysis. Then it leads to introduce new gene in microbes to produce enzymes, therapeutic proteins and even biofuels. For example, माणसामध्ये इन्सुलिन जीन हा ई कोलायमध्ये ट्रान्सफर करून इन्सुलिन हार्मोनचे उत्पादन केले जाते. This is about the genomics. Now, second point is Human Genome Project. So, what is a Human Genome Project? तर Human Genome Project स्टडी करण्यापूर्वी ईस्ट ड्रोसोफिला माऊस यांचा जीनोम स्टडी केला आहे, ज्याद्वारे या सर्व ऑर्गॅनिझमची तुलना ह्यूमन जीनोम बरोबर करता आली. आता हा प्रोजेक्ट कधी सुरू केला, त्यासाठी कोणकोणत्या कंट्रीजनी त्याच्यामध्ये कॉन्ट्रीब्युशन केलं हे आपण थोडक्यामध्ये बघूया. द ह्यूमन जीनोम प्रोजेक्ट वॉज इनिशिएटेड इन 1990 अंडर द इंटरनॅशनल ॲडमिनिस्ट्रेशन ऑफ द ह्यूमन जीनोम ऑर्गनायझेशन, that is HUGO. This project was coordinated by the US Department of Energy and National Institute of Health. Additional contributors included universities across the United States and international partners in the United Kingdom, France, Germany, Japan, India, and China. The Human Genome Project formally began in 1990 and was completed in 2003. म्हणजे बरोबर त्याला 13 वर्षाचा कालावधी हा ह्युमन जिनोम प्रोजेक्ट कंप्लीट होण्यासाठी लागलेला आहे. The Human Genome Project is a multinational research project to determine the genomic structure of humans. Now, let us study about the main aims of the project. First, mapping the entire human genome at the level of nucleotide sequences. Then, to store the information collected from the project in databases. Then, to develop tools and techniques for analysis of the data. Then, to transfer of the related technologies to the private sectors such as industries, and taking care of the legal, ethical, and social issues which may arise from project. The project was also aimed to sequence the genome of several other organisms such as bacteria, for example E. coli, Cynorhabdilis elegans, then yeast, that is Saccharomyces cerevisiae, Drosophila, fruit fly, plants such as rice and Arabidopsis, then mouse, Mus musculus, etc. Complete genome sequence of these model organisms will be useful for comparative studies that will allow researchers to study gene functions in these organisms. Such studies will lead to a greater understanding of human evolution. Then next we study about the importance of Human Genome Project. This Human Genome Project was closely associated with rapid development of a new area in biology called bioinformatics. The work of Human Genome Project has allowed researchers to begin to understand the blueprint in building and constructing the human genome. As researchers learn more about the functions of genes and proteins, this knowledge will have a major impact in the fields like medicine, biotechnology, and the life science. Human Genome Project was to provide a complete and accurate sequence of 3 billion DNA base pairs that make up the human genome and to find out the estimated number of human genes. Now, about 30,000 genes have been estimated to be present in humans. Now, in next slide, see this data. You can find in case of human, chromosome number 46, estimated gene number 33,000, and estimated size of base pair, it is the 3 billion. There are the various organisms: human, mouse, fruit fly, plant, roundworm, yeast, and bacteria. This chart shows comparative genome sizes of humans and other model organisms. See in the slide. Now, next interesting point is DNA fingerprinting. We know genes present on chromosomes are responsible for determining characters of the organisms as well as for inheritance of characters. Due to recombination of parental and maternal genes, we differ from our parents. These differences also arise due to infrequent mutations which occur during gamete formation. Due to all these factors, every individual has its unique genetic makeup, which may be called as fingerprint. The technique developed to identify a person with the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. DNA रेस्ट्रिक्शन ॲनालिसिसच्या मदतीनं पर्सन आयडेंटिफाय करण्याची जी टेक्निक आहे त्यालाच आपण DNA प्रोफाइलिंग किंवा DNA फिंगरप्रिंटिंग असं म्हणतो. The technique of DNA fingerprinting was first given by British geneticist Dr. Alex Jeffreys in 1984. DNA fingerprinting technique is based on identification of nucleotide sequence present in this wonder molecule. We know about 99% of nucleotide sequence in all persons is same. Only some short sequences of nucleotides differ from person to person. In the population, every person shows unusual sequence of 20 to 100 base pairs which are repeated several times. They are termed as variable number of tandem repeats. Very important. VNTRS. Variable number of tandem repeats. The length of regions having VNTRS is different in each individual, and hence it is the key factor in DNA profiling. Now let us study about the steps involved in DNA fingerprinting. There are about 7 steps. First, isolation of DNA. Second, restriction digestion. Third, gel electrophoresis. Fourth, southern blotting. Fifth, selection of DNA probe. Sixth, hybridization. And seventh, photography. Let us study one by one these steps. First step, isolation of DNA. The DNA must be recovered from the cells or tissues of the body, that is the host. Only small amount of tissue like blood, hair roots, skin, etc. is required. That is for DNA isolation. Small amount of tissues like blood, hair roots or skin, they are required. Then second, restriction digestion. The isolated DNA is treated with restriction enzyme. The role of restriction enzyme is to cut the DNA into small fragments having variable lengths. That is, restriction enzyme is a molecular scissor. This entire phenomenon is called RFLP, that is restriction fragment length polymorphism. Now third step, gel electrophoresis. The DNA samples are loaded for agarose gel electrophoresis under an electric influence. इलेक्ट्रोफोरेसिस टेक्निकच्या सहाय्याने डीएनए सॅम्पल्स agarose जेल वरती लोड केले जातात. The DNA fragments which are negatively charged, they move towards the positive pole. DNA fragments ला निगेटिव्ह चार्ज असतो, ते पॉझिटिव्ह पोल कडे मूव्ह होतात. The movement of these fragments depends on length of the fragment, so that there is the formation of the bands takes place. Double stranded DNA, they splits into single stranded DNA by alkali treatment. Now fourth step is southern blotting. There are the 3 types of the blotting: southern blotting for DNA, western blotting for protein, and northern blotting for RNA. Here we study about the southern blotting. Southern, it is the name of the scientist. The separated DNA fragments are transferred to a nylon membrane or a nitrocellulose filter paper by placing it over the gel and soaking them with filter paper overnight. Fifth step, selection of DNA probe. What is a DNA probe? It is nothing but short sequence of DNA. That is, a known sequence of single-stranded DNA is called DNA probe. How DNA probe is obtained? It is obtained from organisms or prepared by cDNA, that is complementary DNA preparation method. The DNA probe is labeled with radioactive isotopes. Now next step, it is the hybridization. What is the hybridization? That is, probe DNA is added to the nitrocellulose filter paper containing host DNA. आता hybridization कसं होतं? Single stranded DNA probe, it pairs with complementary base sequence of the host DNA strand. As a result, DNA-DNA hybrids are formed on the nitrocellulose paper. Remaining single-stranded DNA probe fragments, they are washed off. Next step or last step, it is the photography. The nitrocellulose filter paper is photographed on an X-ray film by autoradiography. The film is analyzed to determine the presence of hybrid DNA. This is about the steps in DNA fingerprinting. This DNA fingerprinting, it can be well explained with the help of the diagram. आता diagram मध्ये बघा. Diagram मध्ये बघा, तिथे sample collected, then isolation of the DNA takes place, then restriction enzyme added. Restriction enzyme, it acts as the molecular scissor. Fragments of DNA obtained. Then these fragments of DNA, they are separate on the basis of their size with the help of the technique electrophoresis. Now they are exposed with the southern blotting. Southern blotting technique न काय केलेलं आहे, त्या मेम्ब्रेन मध्ये, नायट्रोसेल्युलस मेम्ब्रेन मध्ये ब्लॉटिंग केलं जाते. परत त्याच्यामध्ये प्रोब ऍड केला जातो. आता प्रोब ऍड केल्यानंतर हायब्रिडायझेशन ऑफ DNA टेक्स प्लेस. आता प्रोब म्हणजे काय आहे, तर नोन सिक्वेन्स ऑफ द DNA. आता प्रोब हायब्रिडायझेशन कसं होतंय, तर प्रोब प्लस DNA फ्रॅगमेंट. आता प्रोब म्हणजे काय, नोन सिक्वेन्स आहे, त्याला कॉम्प्लिमेंटरी सिक्वेन्स ज्या DNA फ्रॅगमेंटवर असेल तिथे तो हायब्रिडाइज होतो. हा प्रोब रेडिओ लेबल्ड असेल किंवा एन्झाईम लेबल्ड असेल. मग विथ द हेल्प ऑफ फोटोग्राफी आपल्याला काय दिसणार आहे, रेडिओ लेबल्ड असेल तर फ्लोरोसन्स दिसते आणि एन्झाईम लेबल्ड असेल तर तिथं कलर्ड बँड्स दिसतात. अशा पद्धतीनं ही DNA फिंगरप्रिंटिंग जी प्रोसेस आहे ती एंटायर कंप्लीट होते. आता आपण पुढे बघूया ॲप्लिकेशन्स ऑफ DNA फिंगरप्रिंटिंग. DNA फिंगरप्रिंटिंगचा रोल काय आहे किंवा कशामध्ये ते ॲप्लिकेबल आहे. इन फोरेन्सिक सायन्स DNA fingerprinting is used to solve problems of rape and some complicated murder cases. रेप आणि मर्डर केसेसमध्ये फोरेन्सिक सायन्समध्ये DNA फिंगरप्रिंटिंगच्या साहाय्यानं गुन्हेगाराला शोधून काढलं जाते. Then DNA fingerprinting is used to find out the biological father or mother or both of the child in case of the disputed parentage. चाइल्डच्या संदर्भात जे काही डिस्प्यूट असतील पॅरेंट्समध्ये, बायोलॉजिकल फादर कोण आहे, बायोलॉजिकल मदर कोण आहे, ह्यासाठी DNA फिंगरप्रिंटिंग टेक्निक यूज केली जाते. देन DNA फिंगरप्रिंटिंग इज युज्ड इन पेडिग्री अनालिसिस इन कॅट्स, डॉग, हॉर्सेस अँड ह्युमन. या सगळ्याची ह्युमनची असू दे किंवा कॅट्सची, डॉगची, हॉर्सेसची पेडिग्री अनालिसिस शोधण्यासाठी DNA फिंगरप्रिंटिंगचा उपयोग केला जातो. म्हणजेच त्यांची वंशावळ जी आहे ती शोधण्यासाठी DNA फिंगरप्रिंटिंगचा उपयोग केला जातो. Here we completed today's point. In this way we had studied entire topic molecular basis of inheritance, in which we studied following points: discovery of DNA, then DNA as genetic material by Griffith experiments, Avery MacLeod McCarthy experiment, Hershey Chase experiment. Then DNA packaging in prokaryotes and eukaryotes. Then protein synthesis. In this protein synthesis, transcription and translation. Then next point is the genetic code. Then most interesting concept, operon concept. And in this video, genomics, human genome project and DNA fingerprinting. Students, read this point repeatedly and free to ask any query. Thank you.